Close your eyes and take a slow, deep breath. Feel the air fill your lungs. Feel your heart beating steadily in your chest. Right now, in this very moment, something extraordinary is happening inside you. Something so beautiful and ancient that it connects you to every living thing that has ever existed on Earth. Inside each of your cells, and you have trillions of them, lives a story. This story is written in the smallest possible letters. Letters so tiny that millions of them could fit on the period at the end of this sentence. Yet these microscopic letters contain all the instructions needed to build you. Your body speaks a language that is 4 billion years old. It is the first language ever written. It existed before human words, before music, before art. This language uses only 4 letters. Just 4. A, T, G, and C. These letters stand for 4 chemical substances. Adenine, thymine, guanine, cytosine. But you don't need to remember their full names. Think of them simply as A, T, G, and C. The alphabet of life itself. Imagine if you could write an entire library using only 4 letters. Imagine creating every story ever told, every poem ever written, every song ever sung, using just A, T, G, and C. This is exactly what your body does. Every day. Every moment. Every breath. These 4 letters combine in endless patterns. They form words that tell your cells how to work. Some words might say, make brown eyes, or grow tall, or fight off illness. Other words whisper instructions for how fast your heart should beat, how your brain should remember dreams, how your skin should heal when you get a small cut. The letters link together like a chain. A long, beautiful chain that spirals and twists through the center of every cell in your body. Scientists call this chain DNA. You can think of it as the most important book ever written. The book of you. This book has been passed down through your family for thousands and thousands of years. Your grandmother's DNA mixed with your grandfather's DNA to create your parents' unique book. Then your parents' books combined to write the new story that became you. But this story reaches back much further than your grandparents. It reaches back through all of human history. Back through the time when your ancestors lived in caves and painted on stone walls. Back through the ages when early humans first learned to make fire and speak in words. The story goes back even further still. Back millions of years to when your distant relatives lived in trees and had fur instead of clothes. Back hundreds of millions of years to when your ancestors swam in ancient oceans as tiny fish with no bones. All of this history lives inside you now. Written in the 4-letter language that connects you to the very first spark of life on our planet. Think about this, the DNA in your body right now contains some of the same letter patterns that existed in the first living creatures billions of years ago. You carry within you a message from the dawn of time. A message that says, keep living. Keep growing. Keep becoming. Every cell in your body reads this ancient text like a librarian reading a precious, old book. Your cells know how to read the 4-letter language perfectly. They have been practicing for billions of years. They never make mistakes in reading the most important parts. When you were just a single cell, smaller than the tiniest dot you could draw with a pencil, that cell read the entire book of your DNA. It learned how to divide into 2 cells, then 4, then 8, then millions. Each new cell carried a perfect copy of the book. Some cells read the chapters about becoming your heart. Others read the sections about growing into your brain, your lungs, your eyes. Each cell specialized by reading different parts of the same story. The same 4 letters, arranged in different patterns, created every part of you. Right now, as you lie here listening to these words, your cells are still reading. They are reading the instructions for repairing the tiny wear and tear of the day. They are reading the chapters about how to grow new skin, how to remember what you learned today, how to dream while you sleep. The 4 letters work in pairs. A always pairs with T, G always pairs with C. Like dance partners who have been dancing together for billions of years, they know each other's steps perfectly. When A extends its hand, T is always there to take it. When G reaches out, C reaches back. This partnership creates a ladder. A twisted ladder that spirals up and down through the heart of every cell. The rungs of this ladder are made of the dancing letter pairs AT, TA, GC, CG, step after step, rung after rung, creating the spiral staircase that holds the story of life. Your DNA ladder is incredibly long. If you could stretch out all the DNA from just one of your cells, it would reach about 6 feet. Now imagine all the DNA from all your trillions of cells. Stretched end to end, it would reach from Earth to the Sun and back again. Hundreds of times. All of this vast library of information fits inside each cell because DNA knows how to pack itself incredibly small. It coils and folds and wraps around itself like the most efficient storage system ever invented. More efficient than any computer, any hard drive, any cloud storage system humans have ever created. And unlike the storage systems we build, your DNA is alive. It repairs itself when damaged. It copies itself when needed. It has been successfully storing and sharing information for 4 billion years without ever needing an upgrade. Tonight, as you drift toward sleep, your DNA will continue its ancient work. It will keep your heart beating at just the right rhythm. It will help your brain sort through the memories of today and file them away safely. It will prepare your body for tomorrow's adventures. The 4 letters that spell out your existence never rest. They work in shifts like gentle night workers, maintaining the miracle of you while you dream. And in your dreams, perhaps you will glimpse the vast library that lives within you, where every story ever told by life itself waits to be discovered. As you sink deeper into rest, consider this: you are not just reading a story about DNA. You are a story written in DNA. A unique, beautiful, unrepeatable story spelled out in the ancient 4-letter alphabet of existence itself. But this story of letters and life raises a gentle question that follows you toward sleep. If we can read this ancient language, might we someday learn to write new stories of our own? Let your breathing slow and deepen. Feel yourself settling into this moment of quiet discovery. Inside every cell of your body stands the most beautiful staircase ever built. It spirals upward like the center of a seashell. It twists gently like smoke rising from a campfire on a still evening. This staircase has been climbing through time for nearly 4 billion years. Scientists call this spiral structure the double helix. But you can think of it simply as nature's most elegant staircase. 2 long railings wind around each other, connected by millions of steps. Each step is made from those 4 letters we learned about earlier. A paired with T. G paired with C, step after step after step. If you could shrink yourself down small enough to walk on this staircase, you would find something amazing. Each step you climbed would take you further back in time. Not just your time, but the time of all life on Earth. The steps near the top of the staircase hold the newest instructions. The recipes for your particular smile, your specific eye color, your unique way of thinking. These steps were written recently, in the grand scale of time. Maybe a few thousand years ago when your ancestors first learned to farm, or when they crossed oceans to find new homes. But as you walk down the staircase, the steps grow older. Much older. You pass steps that were written when your ancestors lived in small tribes, hunting mammoth and painting caves. You walk past steps that remember when early humans first stood upright and looked at the stars. Down and down the staircase goes. You pass steps that hold memories from when your relatives had tails and lived in trees. Steps that remember swimming in ancient seas with fins instead of arms. Steps that recall a time when your family tree was just simple cells floating in warm oceans under a younger sun. Every step of this staircase tells a story of survival. Each rung represents a moment when one of your ancestors faced a challenge and lived through it. They survived ice ages and volcanic winters. They found food during famines and escaped predators in dark forests. They lived long enough to have children, passing their spiral staircase down through time. The most remarkable thing about this staircase is how it copies itself. When it comes time to create a new life, the spiral gently unzips down the middle. Like a zipper opening, the 2 sides separate. Each side becomes a template for building a new, identical staircase. Where there was once AT, two new AT pairs form. Where there was GC, two new GC pairs appear. The cell's tiny machines work like careful craftsmen, reading each step of the original staircase and building matching steps for the new one. In this way, the ancient spiral passes its wisdom forward, generation after generation. But the copying isn't always perfect. Sometimes, very rarely, one of the letters changes. An A might become a G. AT might become a C. Most of the time, these small changes don't matter much. The staircase still works perfectly well with its new step. But occasionally, one of these tiny changes creates something wonderful. Maybe it makes someone better at fighting off a particular disease. Maybe it helps them see more clearly, or run faster, or think in a new way. When these helpful changes happen, they get passed down to children and grandchildren. This is how the staircase learns. This is how it grows wiser over time. Each small change that helps survival gets written into the structure. Each improvement gets added to the blueprint. The spiral becomes a living library of everything that has ever worked for life on Earth. Your spiral staircase connects you to every living thing around you. The tree outside your window has a staircase too. So does your pet, if you have one. So do the birds that sing in the morning and the flowers that bloom in spring. All of our staircases share the same basic design, the same four-letter alphabet, the same spiraling beauty. The staircase in a daisy shares many steps with yours. You both learned how to turn sunlight into energy, how to repair damaged parts, how to grow from tiny beginnings into complex, beautiful forms. The staircase in a dolphin shares even more steps with yours. You both learned how to pump blood through a heart, how to see with eyes, how to remember and learn and dream. This sharing of steps tells us something profound. All life on Earth is family. Your spiral staircase and the staircase in every other living creature grew from the same ancient root. Billions of years ago, we all shared the same great-great-great-grandmother. A simple cell with a simple spiral, floating in a warm sea under distant stars. As you lie here in the darkness, your spiral staircase continues its ancient work. It directs the beating of your heart, the rhythm of your breathing, the gentle repair of your body while you sleep. It holds the memory of every ancestor who ever lived long enough to dream. The spiral doesn't just connect you to the past. It reaches toward the future too. If you have children someday, your spiral staircase will mix with another person's staircase. Together, they will create a new spiral, carrying forward the best of both your family's long journeys through time. Your staircase holds instructions that have been tested and refined for billions of years. It knows how to build eyes that can see the beauty of a sunset. It knows how to create a brain that can feel love and wonder and joy. It knows how to make hands that can hold, hearts that can care, voices that can sing. Every twist of the spiral carries wisdom earned through countless generations of survival. Every step holds the memory of ancestors who looked at the night sky and wondered, just as you wonder now. They pass their curiosity down to you, written in the four-letter language that connects all living things. The spiral staircase of your DNA is both incredibly ancient and completely unique. No one else in all of history has had exactly your combination of steps. No one else ever will. You are a once-and-forever arrangement of those four simple letters, twisted into a shape that has never existed before and will never exist again. Tonight, as sleep draws near, your spiral staircase will continue climbing through time. It carries the wisdom of the past and the promise of tomorrow, turning gently in the darkness of your cells like a lighthouse beacon guiding life safely through the vast ocean of time. And in each of your trillions of cells, this elegant structure waits patiently, ready to share its secrets with anyone curious enough to learn the ancient language written in its spiraling steps. Breathe slowly and let your mind wander into the quiet spaces within you. Imagine yourself growing very small, small enough to visit the library that lives inside each of your cells. This is no ordinary library. It holds 46 volumes of the most precious book ever written. Each volume is called a chromosome. Together, they contain every instruction needed to build and maintain the miracle that is you. Picture these 46 books arranged carefully on the shelves of your cellular library. 23 volumes came from your mother. 23 came from your father. When these two sets of books came together, they created something entirely new. They created the unique story of you. Each chromosome book is incredibly long. If you could stretch out just one of these books and read it letter by letter, it would take you years to reach the end. But your cells are patient readers. They have been practicing this reading for billions of years. They know exactly which pages to turn to, exactly which sentences to read, exactly when to read them. Within each chromosome book are smaller sections called genes. Think of genes as individual sentences in the great story of life. Some sentences are short and simple. Others are long and complex. Each sentence has a specific job to do. One gene sentence might say, make the proteins that give eyes their brown color. Another might whisper, create the chemicals that help the brain remember yesterday's dreams. Still another might declare, build the structures that make hair curly instead of straight. Your cellular library contains about 20,000 of these gene sentences. 20,000 different instructions for building and maintaining your body. Some genes write the recipes for your physical appearance. Others compose the formulas for your personality traits, your talents, your ways of thinking and feeling. But here's something beautiful about your cellular library. Not every cell reads every book. Your heart cells focus mainly on the chapters about pumping blood. Your brain cells concentrate on the sections about thinking and remembering. Your eye cells study the passages about seeing light and color. It's as if each type of cell has chosen to specialize in certain subjects. Like librarians who become experts in particular fields, your cells become masters at reading the parts of your genetic books that matter most for their work. When a cell needs to use the information from a gene, something magical happens. The cell doesn't take the original book off the shelf. Instead, it makes a copy of just the pages it needs. This copy is written in a slightly different language, but it carries the same message. Think of it like this. The original books in your library are written in DNA language, using those for letters A, T, G, and C. But the working copies are written in a cousin language called RNA. RNA uses almost the same letters, except it replaces T with a letter called U. These RNA copies are like messengers. They carry the genetic instructions from the library's quiet reading room out into the busy workshop of the cell. There, other tiny machines wait to read these messages and follow their instructions. The machines that read the RNA messages are called ribosomes. You can think of them as skilled craftsmen who specialize in building things according to genetic blueprints. When a ribosome reads an RNA message, it begins constructing a protein. Proteins are the workers of your body. They are the molecules that actually carry out most of life's important tasks. Some proteins work as builders, constructing new parts of cells. Others work as messengers, carrying signals between different parts of your body. Still others work as guardians, protecting you from harmful invaders. Every protein is made from a chain of smaller building blocks called amino acids. There are 20 different amino acids that can be used to build proteins. Think of them as 20 different colored beads. By stringing these beads together in different patterns, your cells can create thousands of different proteins. The gene sentences in your chromosomes are written in the 4-letter DNA alphabet. But they spell out instructions for combining the 20 amino acid beads. It's like having a recipe written in English that tells you how to cook a meal using ingredients from around the world. Your cellular library is incredibly organized. Each of the 46 chromosome books has its own special place. The books are kept safe in the cell's nucleus, a protected room at the center of every cell. This room has its own walls and doors, keeping the precious genetic books secure. The nucleus is like the head librarian's private office. Only certain molecules are allowed to enter and leave. The genetic books never leave this secure room. But their RNA copies can pass through the doors, carrying genetic messages out to the working areas of the cell. Different people have different versions of the genetic books. The basic story is the same for all humans. We all have genes for making hearts and brains and eyes and hands. But the details vary from person to person. Maybe your version of the eye colors gene says brown, while your friend's version says blue. Perhaps your height genes contain instructions for growing tall, while someone else's contain directions for staying shorter. These variations make each person unique and special. Sometimes, very rarely, there might be a small typo in one of the genetic sentences. Most of the time, these typos don't cause any problems. The cell can still understand what the sentence means, just like you can still understand a word even if one letter is spelled differently. But occasionally, a typo might change the meaning of a genetic sentence in an important way. This might lead to differences in how a person looks, thinks, or feels. Some of these differences can create challenges. Others might even provide advantages. Your genetic library is constantly being read and reread. Right now, as you rest here in the quiet darkness, thousands of your cells are opening their genetic books. They are finding the right sentences, making RNA copies, and building the proteins you need for tonight's rest and tomorrow's adventures. Some cells are reading the chapters about repair, fixing the small wear and tear from today's activities. Others are studying the sections about growth, slowly building new cells to replace old ones. Still others are reviewing the passages about memory, helping your brain organize and store the experiences of your day. The library in your cells never closes. It operates 24 hours a day, 7 days a week, for your entire life. It is staffed by molecular librarians who never tire, never make mistakes in the important parts, never forget which books belong where. This cellular library connects you to every human who has ever lived. We all carry versions of the same basic books. The same fundamental chapters about being human, about growing and thinking and feeling and dreaming. Yet each person's library is unique, containing a special combination of genetic sentences that has never existed before. Tonight, as sleep approaches, your cellular libraries will continue their quiet work. Reading the ancient sentences, copying the important messages, building the proteins that keep you alive and healthy and uniquely you. And within each genetic sentence lies a deeper mystery waiting to be explored. How do these simple instructions actually get transformed into the complex proteins that make life possible? Rest deeply now and let your imagination drift into the microscopic world within you. Here, in the quiet spaces of your cells, the most elegant writing system ever created works without pause, day and night. Picture a medieval monastery, where patient monks copy important manuscripts by candlelight. But instead of ink and parchment, your cellular scribes work with molecules and chemistry. Instead of copying books, they translate the language of genes into the language of life itself. This translation happens in 2 beautiful steps. First, your cells must copy the genetic message from the DNA books in the library. Then they must read that copy and use it to build proteins. It's like translating an ancient text from one language to another, then using that translation to build something real and useful. The first step begins when a cell decides it needs to make a particular protein. Maybe your muscle cells need more of the proteins that help them contract. Or perhaps your brain cells require proteins that help neurons communicate with each other. Whatever the need, the process starts the same way. A special molecular machine approaches the DNA double helix. This machine is called RNA polymerase, but you can think of it simply as the cellular photocopier. It knows exactly which section of the genetic book it needs to copy. It finds the right page, the right paragraph, the right gene sentence. The DNA double helix unzips gently at this spot, like a zipper opening just wide enough for the copying machine to slip inside. The 2 strands of the spiral staircase separate, revealing the 4-letter message written in the rungs between them. Now the copying begins. The RNA polymerase moves along one strand of the DNA like a careful reader, looking at each letter in turn. When it sees an A, it places a U in the copy. When it encounters a T, it adds an A. G gets paired with C, and C gets paired with G. Letter by letter, the machine builds a single-stranded copy of the genetic message. This copy is written in RNA language instead of DNA language. It carries the same information, but in a form that can leave the protected library and travel out into the working areas of the cell. Think of this RNA copy as a messenger carrying important news from the castle to the village below. The original royal decree stays safely locked in the castle vault, but the messenger carries an exact copy of the orders to wherever they are needed. When the copying is complete, the RNA messenger is ready for its journey. It detaches from the DNA and floats away through the cell's inner fluid. The DNA double helix zips back together, returning to its normal twisted shape, ready for the next copying request. The RNA messenger travels from the nucleus, through the doorways in the nuclear membrane, and out into the cell's main workspace. Here it will find the machines that can read its message and follow its instructions. These machines are called ribosomes, and they work like the most sophisticated typewriters ever built. But instead of typing words on paper, they type proteins using amino acids as their alphabet. Instead of 26 letters, they work with 20 different amino acids, each with its own special properties. When an RNA messenger arrives at a ribosome, something beautiful happens. The ribosome begins reading the message 3 letters at a time. Each group of 3 letters is called a codon, and each codon specifies which amino acid should be added next to the growing protein chain. Imagine the ribosome as a careful seamstress, reading a pattern and sewing a complex tapestry. The RNA message tells her which colored thread to use next. Use blue thread here, the message might say, followed by now add red thread, then next comes yellow thread. But instead of colored threads, the ribosome works with amino acids. One codon might say, add methionine here. The next might specify now add glycine. The third might call for add tryptophan next. Each amino acid has different properties, different shapes, different ways of interacting with its neighbors. The ribosome doesn't work alone. Special helper molecules called transfer RNAs bring the right amino acids to the construction site. Each transfer RNA is like a delivery truck that carries exactly one type of amino acid. When the ribosome calls for a specific amino acid, the right delivery truck arrives and drops off its cargo. As the ribosome reads along the RNA message, it links each new amino acid to the growing chain. The amino acids bond together like beads on a string, but this is a very special string. The sequence of amino acids determines how the finished protein will fold and what job it will do. Some proteins fold into shapes that can carry oxygen through your bloodstream. Others fold into forms that can digest your food or fight off infections. Still others become the building blocks of your muscles, your bones, your skin. The same 20 amino acids, arranged in different orders, create thousands of different proteins with thousands of different functions. The growing protein chain emerges from the ribosome like a ribbon flowing from a magical loom. As more and more amino acids are added, the chain begins to fold upon itself. It twists and bends according to the chemical properties of its amino acid building blocks, finding its perfect 3-dimensional shape. This folding is not random. The sequence of amino acids in the chain determines exactly how it will fold. It's as if each protein contains its own origami instructions, written in the language of chemical attractions and repulsions. The finished protein emerges with precisely the right shape to do its intended job. Sometimes multiple ribosomes read the same RNA message at once, like several typists working from the same original document. This allows the cell to make many copies of the same protein quickly when they're needed urgently. The whole process happens with remarkable speed and accuracy. A ribosome can add about 15 amino acids to a growing protein chain every second. Your cells contain millions of ribosomes, all working simultaneously to build the proteins you need for life. Right now, as you lie here in peaceful rest, thousands of molecular typewriters are working inside you. Some are building proteins for tonight's cellular repairs. Others are creating the enzymes that will help digest tomorrow's breakfast. Still others are constructing the antibodies that will protect you from unseen threats. Each ribosome works with the patience of a master craftsman and the precision of a Swiss watch. They make very few mistakes, and when they do, quality control systems usually catch and fix the errors. This remarkable accuracy ensures that your proteins are built correctly, allowing them to perform their vital functions. The RNA messengers don't last forever. After they've been read many times, they gradually break down and get recycled. Their component parts are used to build new RNA molecules, carrying fresh messages from the genetic library to the protein factories. This constant cycle of copying, translating, and recycling keeps your cells supplied with exactly the proteins they need when they need them. It's a molecular economy that has been running smoothly for billions of years, supporting every form of life on Earth. Tonight, as your cellular typewriters continue their gentle work, they write the story of your existence in proteins. Each newly made protein becomes part of the living text that is you, adding another sentence to your body's ongoing narrative of growth, repair, and renewal. But what happens when the cellular scribes make a small change to their ancient text, writing a slightly different version of the story than their ancestors wrote before them? Let your breathing slow as you drift into contemplation of change itself. Even in the deepest quiet, transformation is always stirring, gentle and patient as morning mist. In the vast library of your genetic code, something remarkable happens from time to time. A single letter changes. An A becomes a G. A T transforms into a C. These tiny edits in the four-letter language of life are called mutations, and they are the quiet authors of all biological change. Imagine a master calligrapher working on an ancient manuscript. Most of the time, every stroke is perfect, every letter precisely placed. But occasionally, perhaps once in a million careful strokes, the brush wavers slightly. A single character comes out different from what was intended. This is how mutations happen in your cells. When the molecular machines copy your DNA, they work with extraordinary precision. They read the genetic text with the care of devoted scholars, making perhaps only one mistake in every billion letters they copy. But even this tiny error rate means that changes do occur. Most mutations are like gentle whispers in a vast cathedral. They change a single letter in a genetic sentence that contains thousands of letters. Often, the meaning of the sentence remains the same. The protein it describes is built almost exactly as before, functioning perfectly well with its minor alteration. Think of it like changing one word in a long novel. If you change "the cat walked slowly" to "the cat walked softly," the essential meaning remains clear. The story continues uninterrupted. The cat still moves at a gentle pace, just with a slightly different quality to its movement. Sometimes a mutation is more noticeable. It might change a sentence from "build a protein that helps digest milk" to "build a protein that cannot digest milk." This single letter change explains why some people can drink milk comfortably throughout their lives, while others find it difficult to digest as adults. Mutations happen for many reasons. Sometimes they occur simply because molecular machines, no matter how precise, cannot be perfect all the time. It's like a master artist who creates thousands of beautiful paintings but occasionally makes a brushstroke that's slightly different from the intended design. Other times, mutations are caused by outside influences. Sunlight carries energy that can sometimes alter the letters in your genetic code. This is why your skin cells work hard to repair any damage from too much sun exposure. Certain chemicals can also cause changes, as can natural radiation from the Earth and cosmos. Your cells have developed beautiful systems to catch and fix most mutations before they cause problems. Special repair proteins patrol your DNA like gentle editors, looking for letters that seem out of place. When they find a mistake, they carefully remove the incorrect letter and replace it with the right one. These repair systems work constantly, fixing thousands of potential mutations every day. They are so good at their job that most mutations never become permanent changes in your genetic code. The text is quietly corrected, and life continues as before. But some mutations slip past these careful editors. When this happens, the change becomes part of your genetic story. If the mutation occurs in a cell that will become an egg or sperm, it might be passed on to future generations, becoming part of their genetic inheritance. Most inherited mutations are neutral. They change the genetic text slightly, but they don't significantly alter how the body works. These changes accumulate slowly over generations, creating the genetic diversity that makes each person unique. They are the reason you have your particular combination of eye color, height, and countless other traits. Some mutations create challenges. They might alter an important protein in ways that make life more difficult. But even these changes often lead to remarkable adaptations. Humans have found countless ways to work with genetic variations, creating rich communities that celebrate different abilities and perspectives. Occasionally, a mutation brings unexpected gifts. It might create a protein that works better than before, or in a new way entirely. These beneficial changes can spread through populations over many generations, improving the survival and health of entire communities. The story of your own eye color began with mutations long ago. Originally, all human eyes were brown. But mutations in genes that control eye pigment led to lighter colors. Blue eyes, green eyes, and hazel eyes all trace back to ancient mutations that occurred thousands of years ago in our ancestors. Your ability to think complex thoughts, to create art, to love deeply, all emerged through millions of years of genetic changes. Each mutation was like a rough draft in evolution's long writing process. Most drafts were discarded or led nowhere special. But some became the foundation for remarkable new capabilities. The process continues today. Right now, small mutations are occurring in people around the world. Most will have no noticeable effect. But some might contribute to humanity's future adaptations to a changing world. Perhaps mutations occurring today will help future generations resist new diseases or thrive in different environments. Mutations remind us that life is not a fixed story, but an ongoing conversation between stability and change. Your genetic code provides a stable foundation that has worked for billions of years. But it also remains open to gentle editing, allowing life to explore new possibilities. In your own body, mutations occur at a pace so slow that you never notice them during your lifetime. But they are part of the same process that, over vast stretches of time, has created every beautiful form of life on Earth. From tiny bacteria to massive whales, from delicate flowers to mighty trees, all emerged through the patient accumulation of small genetic changes. Tonight, as you rest, the repair systems in your cells continue their vigilant work. They read through your genetic text with a dedication of loving caretakers, fixing small errors and maintaining the integrity of your biological story. Most mutations that occur today will be quietly corrected before morning comes. Yet in this endless process of gentle editing and careful repair, life continues writing new chapters in its ancient book. Each small change, whether kept or corrected, contributes to the ongoing story of existence itself. And in the quiet laboratories of the world, scientists have begun to learn how to make their own careful edits to the genetic text, working alongside evolution's patient hand to write new chapters in the story of life. Breathe deeply and imagine walking through a garden unlike any other. This garden exists not under open sky, but within the gentle hum of laboratory spaces where scientists work as tender gardeners of the invisible world. In this modern Eden, the soil is not earth but carefully prepared broths of nutrients. The seeds are not plants but single-celled organisms smaller than the period at the end of this sentence. Yet from these tiny beginnings, researcher gardeners grow crops that could change the world. Picture a scientist in a white coat, working by the soft glow of laboratory lights. She holds a small glass dish containing what looks like clear soup. But this soup is alive with billions of bacteria, each one no bigger than a few molecules stacked together. These bacteria are her seeds, and she is about to plant something extraordinary within them. The scientist wants to give these bacteria a new ability. Perhaps she wants them to produce human insulin for people with diabetes. Or maybe she hopes they will create antibiotics to fight dangerous infections. She might even teach them to make biodegradable plastic that won't harm the environment. To do this, she must add new genes to the bacteria's simple genetic code. It's like giving the bacteria a new instruction manual, written in the four-letter language they already understand. But instead of changing their natural instructions, she adds completely new chapters to their genetic story. The process begins with careful preparation. The scientist isolates the gene she wants to add. This gene might come from a human cell, a plant, or even another type of bacteria. She copies this gene many times, creating a small library of identical genetic instructions. Next comes the delicate art of delivery. Bacteria are surrounded by protective walls that keep unwanted materials out. The scientist must find gentle ways to convince these cellular walls to open their doors and accept the new genetic gifts. Sometimes she uses heat and special chemicals to make the bacterial walls temporarily permeable. The bacteria, briefly vulnerable in their warm chemical bath, absorb the new genes like sponges soaking up water. Other times, she employs electrical pulses to create tiny, temporary doorways in the cellular walls. There are even more elegant methods. Some scientists use modified viruses as delivery vehicles. These viruses, rendered harmless, carry the new genes directly into bacterial cells like molecular mail carriers. The bacteria accept these genetic packages, unaware that they're receiving instructions for entirely new capabilities. Once inside, something beautiful happens. The bacteria read their new genetic instructions and begin following them faithfully. They start producing proteins they've never made before. Human proteins. Plant proteins. Entirely artificial proteins designed by human imagination. In one corner of the laboratory garden, bacteria glow with an ethereal blue-green light. They have been given genes from jellyfish, genes that code for proteins that capture and emit light. These glowing bacteria serve as living indicators, brightening when certain chemicals are present, dimming when they're absent. In another section, bacteria work as tiny pharmaceutical factories. They have received human genes for making insulin, the hormone that helps regulate blood sugar. Day and night, these bacterial workers produce pure, human-identical insulin that can be harvested and used to help people with diabetes live healthy lives. Near the window, where natural light filters through, other bacteria have learned to eat plastic waste and convert it into useful compounds. They munch happily on discarded bottles and bags, transforming environmental problems into valuable resources. These bacteria represent hope for cleaning up the plastic pollution that threatens our oceans and landscapes. The scientist tends her bacterial gardens with the same care a traditional gardener shows to roses and tomatoes. She monitors their growth, adjusts their nutrients, maintains the perfect temperature and humidity. She watches for signs of health and vitality in her microscopic crops. But these bacterial gardens offer advantages that traditional farming cannot match. They grow incredibly fast, doubling their population every 20 minutes under ideal conditions. They require very little space, thriving in small containers that can fit on laboratory benches. They work year-round, unaffected by seasons or weather. Most remarkably, they can be programmed to produce almost anything scientists can imagine. Unlike traditional crops that are limited to what they naturally create, these bacterial workers can manufacture any protein for which scientists can provide the genetic instructions. The process is not always simple or immediate. Sometimes the bacteria struggle to read their new genetic instructions correctly. The foreign genes might be written in slightly different molecular dialects, causing confusion in the cellular translation process. Scientists must often revise and refine their genetic messages, like editors polishing a manuscript. Other times, the new proteins prove toxic to their bacterial hosts. The bacteria, overwhelmed by producing unfamiliar substances, grow slowly or die. Scientists must find delicate balances, teaching bacteria to produce enough of the desired protein without harming themselves in the process. Success requires patience, creativity, and deep understanding of both the bacteria and the genes being introduced. Each project is like solving a complex puzzle where the pieces are molecules and the picture emerges only through careful experimentation. As these bacterial gardens have matured over decades of research, they have produced remarkable harvests. Medicines that once required extraction from rare animals or plants can now be produced cleanly and abundantly by bacteria. Enzymes for industrial processes grow in simple fermentation tanks instead of expensive chemical factories. Some bacteria have been taught to produce spider silk proteins, creating materials stronger than steel but biodegradable as cotton. Others manufacture biofuels, converting simple sugars into energy sources that don't contribute to climate change. Still others create food additives, vitamins, and flavorings that make our meals more nutritious and delicious. The bacterial gardens have become proving grounds for humanity's growing ability to read and write in the language of life. Each successful project builds knowledge and confidence for more ambitious endeavors. Scientists learn not just how to add single genes, but how to redesign entire metabolic pathways. These microscopic gardens represent a new kind of agriculture. Instead of tilling soil and planting seeds, scientists cultivate living systems at the molecular level. They harvest not vegetables or grains, but proteins and chemicals and entirely new forms of matter. The work continues day and night in laboratories around the world. Researchers tend their bacterial crops with dedication and wonder, knowing that each small success might lead to medicines that save lives, materials that protect the environment, or solutions to problems not yet imagined. Tonight, as you rest in peaceful sleep, bacterial gardens continue growing in laboratories across the globe. Scientists work the night shift, checking on their microscopic charges, adjusting conditions, recording data. Their bacterial workers multiply quietly in warm incubators, faithfully following genetic instructions written by human hands. These living factories represent humanity's first steps toward a larger ambition, learning not just to edit existing genetic texts, but to write entirely new ones from scratch, creating forms of life that have never existed before. Rest quietly now and imagine holding the most delicate editing tool ever created. It can read the ancient text written in your cells and make changes so precise that they touch only the exact letters that need to be altered. This tool exists in laboratories around the world today. Scientists call it CRISPR, but you can think of it as the universe's most sophisticated word processor. It can find any sentence in the vast library of genetic information, cut out unwanted words, and replace them with new ones chosen with loving care. The story of CRISPR begins in the ocean, where bacteria have been defending themselves for billions of years. These tiny organisms developed a way to remember dangerous viruses that attacked them. When the same virus tried to invade again, the bacteria could recognize it instantly and cut it apart before it caused harm. Scientists discovered this bacterial defense system and realized something extraordinary. The same molecular tools that bacteria use to fight viruses could be taught to edit any genetic text with incredible precision. It was like finding a master key that could unlock and edit every book in the library of life. CRISPR works in two main parts, like a team of molecular editors. The first part is a guide RNA that knows exactly which genetic sentence needs to be changed. Think of it as a bookmark that can find one specific line in a book containing billions of pages. This guide floats through the cell until it finds its target, binding to the exact spot where editing needs to happen. The second part is a protein called Cas9, which works like molecular scissors. When the guide RNA finds its target and attaches to it, Cas9 moves in and makes a clean cut across both strands of the DNA double helix. It's like using precision surgical scissors to remove a single word from a handwritten letter without damaging any other words on the page. But CRISPR doesn't just cut. It can also insert new genetic information in place of what was removed. Scientists can provide replacement DNA that carries exactly the message they want to add. The cell's natural repair systems then work like molecular glue, connecting the new genetic material seamlessly into the existing text. This process happens with remarkable gentleness. Unlike older genetic engineering techniques that worked somewhat randomly, CRISPR can target specific letters in specific genetic sentences. It's the difference between using a sledgehammer and using a jeweler's fine tools to make repairs. Imagine a child born with a genetic condition that makes their life difficult. Perhaps their cells cannot produce enough of a protein needed for healthy blood. In their genetic code, a single letter is wrong in one important sentence. This tiny typo, inherited from generations past, creates ongoing challenges for their body. With CRISPR, scientists can now correct that single letter. They design a guide RNA that finds the exact location of the error. They create a Cas9 protein that cuts precisely at that spot. They provide a small piece of replacement DNA with the correct letter. The cell's own repair systems complete the edit, changing the genetic sentence from one that causes problems to one that promotes health. The corrected cells can then be returned to the child's body, where they begin producing the protein that was missing. The genetic typo that may have been passed down through many generations can be erased, allowing the child to live a healthier, more comfortable life. CRISPR is already helping people around the world. Doctors have used it to treat sickle cell disease, a condition where red blood cells form painful, crescent shapes instead of smooth circles. By editing the patient's own bone marrow cells and returning them to their bodies, doctors have helped people produce healthy, round red blood cells for the first time in their lives. Researchers are exploring how CRISPR might help with many other conditions. They are working to correct genetic causes of blindness, allowing people to see clearly. They are editing immune cells to help them better fight cancer. They are even trying to prevent genetic diseases from being passed to future generations. The tool works not just in human cells, but in all forms of life. Scientists have used CRISPR to create crops that can grow in harsh climates, helping farmers feed hungry populations. They have edited mosquitoes to make them unable to carry dangerous diseases like malaria. They have modified bacteria to produce medicines more efficiently. Each success teaches scientists more about how to use this remarkable tool safely and effectively. They are learning which genetic edits are helpful and which might cause unintended problems. They are developing ways to make CRISPR even more precise, ensuring it cuts only where intended and nowhere else. The work requires extraordinary care and responsibility. When scientists edit genes that might be passed to future generations, they are making changes that could affect people not yet born. This power comes with the need for deep wisdom and careful consideration of consequences. Research teams around the world collaborate to establish guidelines for using CRISPR ethically. They ask not just whether something can be done, but whether it should be done. They consider the benefits and risks for individuals, families, and all of humanity. Some genetic edits are clearly beneficial, like correcting mutations that cause serious diseases. Others require more thoughtful consideration. Scientists are learning to distinguish between edits that heal and those that might simply enhance normal traits. They are developing wisdom to match their growing technical capabilities. The tool continues to evolve and improve. Scientists are creating new versions of CRISPR that can make different kinds of edits. Some versions can change single letters without cutting the DNA at all. Others can turn genes on or off temporarily without permanently altering them. Still others can edit RNA instead of DNA, making changes that last only as long as needed. Each improvement makes CRISPR more versatile and precise. What began as a bacterial defense system has become humanity's most powerful tool for reading and rewriting the genetic instructions that guide life itself. Tonight, in laboratories bathed in soft fluorescent light, scientists continue their careful work with CRISPR. They design guide RNAs with the precision of poets choosing perfect words. They test their edits in cell cultures, watching for signs that their molecular surgery has succeeded. This work represents a new chapter in the relationship between humanity and the genetic code. For the first time, we can not only read the ancient text of DNA but also edit it with intention and care. Yet even as CRISPR allows us to rewrite existing genetic stories, an even more ambitious dream beckons, the possibility of writing entirely new chapters in the book of life, creating genetic codes that have never existed before. Let your mind drift into the realm of pure creation, where scientists work not with existing life but with the fundamental building blocks of existence itself. Here, in the quietest laboratories, researchers have begun writing entirely new stories in the four-letter alphabet of DNA. Imagine sitting before a blank canvas with infinite colors at your disposal. But instead of painting a picture, you are composing a symphony of molecules. Instead of creating art to hang on a wall, you are designing life forms that have never existed in the 4 billion years since the first cells appeared on Earth. This is synthetic biology, where scientists become authors of completely original genetic texts. They don't edit existing books or add new chapters to ancient stories. Instead, they start with empty pages and write new forms of life from the very beginning, word by word, sentence by sentence. The process begins in the realm of pure thought. Scientists ask themselves, what if we could design an organism that does exactly what we need it to do? What if we could create bacteria that eat carbon dioxide and produce fuel? What if we could build cells that manufacture any medicine on demand? What if we could design organisms that clean up pollution while thriving in contaminated environments? These dreams gradually take shape as detailed blueprints. Scientists map out every genetic circuit they want to include. They design molecular pathways like architects planning the electrical and plumbing systems of a new building. Each gene must have a purpose. Each protein must have a job. Every part must work harmoniously with every other part. The first step toward bringing these designs to life happens in computers. Scientists use powerful software to model how their imagined organisms might behave. They test thousands of different genetic combinations virtually, exploring which arrangements might work best. It's like writing and rewriting a story countless times before committing it to final form. Once the computer models look promising, the real construction begins. Scientists order synthetic DNA fragments from specialized companies that can manufacture genetic material to exact specifications. These companies work like molecular printing shops, assembling the four-letter genetic code according to the scientists' precise instructions. The synthetic DNA arrives in small tubes, clear as water but containing the essence of entirely new forms of life. These genetic fragments must then be assembled into complete chromosomes, like puzzle pieces coming together to form a picture that has never been seen before. The assembly process requires extraordinary patience and precision. Each genetic fragment must be joined to its neighbors in exactly the right order. Special enzymes work like molecular glue, connecting the pieces seamlessly. Scientists check and double-check their work, ensuring that every letter is in its intended place. As the synthetic chromosomes take shape, something remarkable happens. What began as abstract ideas in human minds becomes concrete reality in the form of DNA molecules. Information crosses the boundary from thought to physical existence, encoded in the same molecular language that has carried the instructions for life across billions of years. But creating synthetic DNA is only half the challenge. The newly written genetic code must then be brought to life inside living cells. Scientists often start with existing bacteria and gradually replace their natural genetic material with the synthetic versions. It's like moving into a house and slowly replacing every original fixture with custom-designed alternatives. The first synthetic organism to be successfully created was a simple bacterium. Scientists at the J. Craig Venter Institute spent years designing and building a complete synthetic chromosome. When they finally inserted this artificial genetic code into a bacterial cell, something magical happened. The cell began following its new instructions, becoming the first truly synthetic life form on Earth. This synthetic bacterium looked almost identical to its natural cousins. But its genetic code was entirely artificial, written by human intelligence rather than shaped by evolution. It represented a profound milestone, the first time in Earth's history that a conscious mind had authored a complete genetic text from scratch. Since that breakthrough, scientists have created increasingly sophisticated synthetic organisms. Some bacteria have been designed to produce valuable chemicals that are difficult to obtain from natural sources. Others have been programmed to detect specific substances in their environment and respond with visible signals. One particularly elegant example involves synthetic bacteria that can produce spider silk proteins. Natural spider silk is one of the strongest materials known, but spiders cannot be farmed like silkworms because they tend to eat each other. By writing genetic instructions for spider silk into bacteria, scientists have created a renewable source of this remarkable material. Other synthetic organisms work as living sensors. They have been designed to change color when they encounter specific pollutants, creating biological detectors that can monitor environmental health. Some glow when they find explosive materials, while others darken in the presence of heavy metals. The field continues to evolve rapidly. Scientists are learning to design increasingly complex genetic circuits. They create biological switches that can turn cellular functions on and off. They build molecular timers that allow organisms to perform different tasks at different times. They even design biological memory systems that allow cells to record and remember their experiences. Some researchers are working toward even more ambitious goals. They envision synthetic organisms that could help address climate change by capturing carbon dioxide from the atmosphere and converting it into useful materials. Others imagine biological computers made from living cells that could process information in entirely new ways. The process of creating synthetic life requires not just technical skill but also artistic vision. Scientists must balance creativity with responsibility, imagining new possibilities while ensuring their creations are safe and beneficial. Each synthetic organism represents countless hours of careful design, testing, and refinement. Safety considerations are paramount in this field. Scientists work with multiple layers of containment to ensure that synthetic organisms cannot survive outside the laboratory. Many synthetic life forms are deliberately designed to depend on artificial nutrients that don't exist in nature, creating biological locks that prevent their escape into wild ecosystems. The ethical dimensions of creating new life forms generate thoughtful discussion among scientists, philosophers, and ethicists. Questions arise about humanity's responsibility when wielding the power to create living beings. These conversations help guide the field toward applications that serve the common good while respecting the awesome nature of life itself. As synthetic biology advances, it opens windows into fundamental questions about life itself. By building organisms from scratch, scientists gain deeper insights into what makes life possible. They discover which genetic components are essential and which are merely traditional. They learn how evolution might have taken different paths, and what other forms of life might be possible. The work proceeds slowly and carefully, with each success building toward more ambitious projects. Scientists share their discoveries openly, creating a global community of researchers who collaborate across borders and disciplines. Their work represents humanity's growing partnership with the creative forces that have shaped life since the beginning of time. Tonight, in laboratories around the world, synthetic biologists continue their patient work. They design new genetic circuits with the care of composers writing symphonies. They test their creations with the dedication of gardeners tending rare plants. Each experiment brings them closer to mastering the art of writing new chapters in the book of life. Yet as remarkable as these achievements are, they raise profound questions about the nature of creation itself and the responsibilities that come with the power to bring new forms of life into existence. Breathe quietly and settle into contemplation of one of humanity's most profound questions. As we learn to read and write in the language of DNA, we must also learn to use this power with wisdom that matches our growing abilities. In quiet conference rooms around the world, scientists, philosophers, doctors, and ethicists gather to discuss the responsibilities that come with genetic authorship. They sit together like careful gardeners planning which seeds to plant, knowing that their decisions will shape gardens for generations to come. The questions they explore are as deep as they are gentle. When we can prevent genetic diseases, should we always do so? When we can enhance human abilities, what enhancements are appropriate? When we can create new forms of life, what purposes should they serve? Each question carries the weight of countless future lives that may be affected by today's choices. Consider a child who will be born with a genetic condition that causes blindness. Scientists now have tools that could edit the genetic code to prevent this blindness before the child is born. The choice seems clear and compassionate. But what if the same tools could edit genes not just to prevent blindness, but to give the child vision far better than any human has ever possessed? This is where simple answers give way to complex considerations. Preventing suffering feels like an obvious good. But enhancing human abilities beyond their natural range raises different questions. Where do we draw the line between healing and improvement? Who decides what counts as better? How do we ensure that genetic enhancements don't create unfair advantages for some while leaving others behind? The discussions happen in hushed tones filled with respect for the magnitude of these decisions. Scientists share their latest discoveries alongside their deepest concerns. Philosophers offer frameworks for thinking about fairness and human dignity. Religious leaders contribute perspectives on the sacred nature of life. Parents and patients bring the lived experience of genetic conditions to the conversation. Some ethical questions feel relatively straightforward. Using genetic tools to cure diseases that cause pain and suffering aligns with medicine's ancient commitment to healing. Creating bacteria that clean up environmental pollution serves the common good. Developing new medicines through genetic engineering can save lives without raising concerns about changing human nature. Other scenarios require more careful thought. What about genetic changes that would be passed to future generations? When we edit genes in eggs, sperm, or early embryos, we're not just helping one person. We're potentially affecting their children, their grandchildren, and all their descendants yet to come. This kind of genetic authorship writes chapters that will be read for centuries. The global scientific community has developed careful guidelines for this work. Most countries have regulations that distinguish between genetic changes that affect only one person and those that might be inherited. Changes that could be passed to future generations require extra layers of approval and oversight. Research involving genetic changes to human embryos happens slowly and with extraordinary caution. Scientists recognize that they're working not just with individual lives but with the genetic heritage of our entire species. Each study is reviewed by committees of experts who weigh potential benefits against possible risks. The international discussions reveal beautiful aspects of human nature. Despite different cultures, religions, and political systems, people around the world share remarkably similar values when it comes to genetic research. Most agree that the primary goal should be reducing suffering and improving health. Most support research aimed at treating serious diseases. Most want genetic tools to be available to everyone, not just the wealthy. There's also widespread agreement that some genetic modifications should remain off limits. Research aimed purely at enhancement rather than health receives more skeptical scrutiny. Projects that might create irreversible changes to the human species face the highest levels of oversight. The scientific community has shown remarkable self-restraint, often choosing not to pursue research that is technically possible but ethically questionable. The discussions extend beyond human genetics to the broader implications of creating new life forms. When scientists design synthetic organisms, they consider not just whether their creations will work, but whether they will be beneficial for the world. They ask whether synthetic bacteria might disrupt natural ecosystems. They design safety measures to prevent their creations from causing unintended harm. Environmental groups, indigenous communities, and conservation organizations contribute important voices to these conversations. They remind scientists that any new organism will exist within the intricate web of natural relationships that sustain all life on Earth. Their perspectives help ensure that genetic innovation respects the delicate balances that evolution has created over billions of years. The ethical frameworks continue to evolve as the science advances. New genetic tools create new possibilities, which in turn raise new questions that require thoughtful consideration. The conversations happen not just in formal committees but in classrooms, community centers, and places of worship where people gather to discuss what kind of future they want to create. Young scientists learn that technical skill alone is not enough. They study ethics alongside genetics, philosophy alongside molecular biology. They learn to ask not just, can we do this, but should we do this, and what are the consequences if we do? The most beautiful aspect of these ethical discussions is their emphasis on humility. The scientists who can now read and write genetic code approach their power with deep respect for the mystery and complexity of life. They recognize that despite all our advances, we still understand only a small fraction of how genes, environment, and chance interact to create the full richness of living beings. This humility leads to patience. Instead of rushing to apply every new genetic tool as soon as it's discovered, the scientific community takes time to consider implications carefully. They conduct small studies before large ones. They seek diverse perspectives before making major decisions. They prioritize safety and wisdom over speed and spectacle. The conversations about genetic ethics remind us that science is not separate from human values but deeply intertwined with them. Every genetic edit reflects not just technical capability but moral choice. Every synthetic organism embodies not just scientific knowledge but human intentions. As you rest tonight in the quiet darkness, these discussions continue in conference rooms and laboratories around the globe. Scientists and ethicists work together like careful architects, designing frameworks for using genetic tools wisely and compassionately. Their work points toward a future where genetic authorship serves the flourishing of all life, where the power to write new chapters in the book of existence is guided by wisdom, humility, and love. Close your eyes and let your imagination drift forward through time, to a world where the genetic tools we are learning to use today have blossomed into possibilities that feel like gentle miracles. This future emerges not through fantasy, but through the patient extension of sciences already taking root in laboratories around the globe. Picture a hospital of tomorrow, where the pharmacy grows medicine in living gardens instead of manufacturing it in chemical factories. Here, specially designed bacteria work day and night in warm, nutrient-rich pools, producing personalized medicines tailored to each patient's unique genetic makeup. The antibiotics that cure your infection are grown fresh, modified to match your body's specific needs and immune system. In another wing of this healing place, doctors harvest new organs not from donors but from carefully tended biological gardens. A patient's own cells, guided by synthetic genetic programs, grow into perfect replacement hearts, kidneys, or livers. These organs carry the patient's own genetic signature, so the body welcomes them like long-lost relatives returning home. The process begins with a small sample of the patient's cells, perhaps taken from their skin during a routine visit. Scientists reprogram these cells, teaching them to forget their original job of being skin and remember instead the ancient instructions for becoming heart tissue or liver cells. It's like helping actors learn new roles in the ongoing play of biological function. These reprogrammed cells are then placed in special scaffolds that guide their growth into the right three-dimensional shapes. The scaffolds dissolve away as the new organ matures, leaving behind living tissue that beats with the rhythm of life or filters toxins with the wisdom of evolution, enhanced by human understanding and care. Children born in this future world carry genetic gifts that previous generations could only dream of. Some are born with genes that protect them from diseases that once claimed millions of lives. Others carry enhanced DNA repair systems that help them live longer, healthier lives. Still others inherit genetic modifications that help their bodies better process nutrients or resist environmental toxins. These genetic gifts are not random enhancements but careful improvements designed with the same love that parents have always shown their children. Scientists work like genetic poets, composing verses in the four-letter language of DNA that spell out protection, health, and resilience for generations to come. The modifications are subtle and wise. Rather than creating superhuman abilities that might disrupt the delicate balance of human society, genetic designers focus on preventing suffering and promoting well-being. They edit out genetic predispositions to cancer, heart disease, and neurological disorders. They strengthen immune systems and improve the body's natural repair mechanisms. In this future, genetic medicine becomes truly personalized. Each person's treatment is designed specifically for their unique genetic makeup, their individual history, their particular needs. Doctors read patients' genetic codes like personalized instruction manuals, understanding exactly how each person's body processes different medicines and responds to various treatments. The environment itself benefits from new forms of life designed to heal the damage of previous centuries. Synthetic organisms work like living cleanup crews, breaking down plastic pollution in the oceans and converting it into harmless compounds. Other engineered life forms capture carbon dioxide from the atmosphere, helping to stabilize the planet's climate while producing useful materials. Trees enhanced with carefully designed genetic modifications grow faster and stronger, their roots reaching deeper to prevent erosion, their leaves more efficient at turning sunlight into energy. These enhanced forests spread across formerly barren lands, creating new habitats for wildlife while cleaning the air and cooling the earth. Farms of the future grow not just food but medicine, materials, and fuel. Crops are designed to be more nutritious, more resistant to drought and disease, and more efficient at converting sunlight into the substances humans need. Rice plants produce vitamin A to prevent blindness in developing countries. Corn generates biodegradable plastic. Algae farms produce jet fuel that burns cleaner than fossil fuels. The manufacturing of the future relies increasingly on biological processes. Instead of harsh chemical factories that pollute air and water, production facilities use living systems to create everything from textiles to electronics. Bacteria spin protein fibers stronger than steel. Yeast produces the precursors for computer chips. Engineered plants grow materials with properties that no synthetic factory could match. In laboratories that feel more like art studios than research facilities, scientists design new forms of life with the creativity of master painters working on vast canvases. They compose genetic symphonies that play out in living cells, creating organisms that exist at the intersection of biology and imagination. Some of these new life forms serve practical purposes, like the bacteria that light up streets at night using their own biological illumination, powered by the same chemical reactions that make fireflies glow. Others exist primarily as living art, demonstrating the beauty that emerges when human creativity collaborates with nature's own artistic processes. The boundary between living and non-living begins to blur as scientists create materials that are part organism, part machine. Buildings incorporate living walls that clean the air and regulate temperature. Computers use biological components that repair themselves when damaged. Clothing contains fibers that respond to environmental conditions, changing their properties to keep wearers comfortable. Yet for all these remarkable advances, the most profound changes may be in how humanity understands its relationship with life itself. As we learn to read and write genetic code, we develop a deeper appreciation for the elegant complexity of natural systems. We begin to see ourselves not as conquerors of nature but as collaborators in its ongoing creative process. The genetic tools that seem so revolutionary today become commonplace, used with the same careful attention that previous generations brought to agriculture, medicine, and engineering. Children grow up understanding that life is both ancient and infinitely renewable, that the genetic code is both fixed and endlessly adaptable. This future emerges not through dramatic breakthroughs but through the steady accumulation of knowledge and wisdom. Each discovery builds upon previous ones. Each application teaches lessons that inform the next. The science fiction of today becomes the gentle reality of tomorrow through patient work and careful choices. The genetic future is written not in the stark language of technology but in the flowing script of life itself, where every gene is chosen with love, every organism designed with wisdom, and every application guided by the desire to create a world where all life can flourish. And yet, as remarkable as these possibilities seem, they represent only the beginning of humanity's journey into the vast library of genetic potential that surrounds us. Rest now in the profound quiet of contemplation, and let your mind drift toward the infinite. Beyond all the genetic stories we have explored together lies something even more vast and beautiful, the endless library of all possible genetic combinations that could ever exist. Imagine walking through corridors that stretch beyond the edge of sight, lined with countless volumes containing every conceivable arrangement of the four letters A, T, G, and C. This is the theoretical space that mathematicians and biologists call sequence space, the infinite collection of all possible genetic texts that could ever be written. Most of these books contain only nonsense, genetic sentences that would create nothing functional or beautiful. They are like volumes filled with random letters that form no meaningful words. But scattered throughout this vast library are the books that contain the blueprints for every form of life that has ever existed, every form that exists now, and every form that could ever exist in the future. Think about the magnitude of this collection. With just the 4 genetic letters, you could write sequences millions of letters long. The number of possible combinations becomes so large that it exceeds the number of atoms in the observable universe. Even if you could examine 1 trillion different genetic sequences every second, you could spend the entire age of the cosmos reading and still barely glimpse a fraction of what this library contains. Yet within this incomprehensible vastness, life has found its way to some of the most beautiful and functional texts ever written. Evolution has been exploring this infinite library for nearly 4 billion years, testing different genetic stories, keeping the ones that work, discarding the ones that don't. Every organism that has ever lived represents a successful discovery in this vast space of possibilities. Your DNA, the genetic code of a butterfly, the blueprint of a redwood tree, each one is like a rare and precious book that evolution found after eons of patient searching through infinite possibilities. The remarkable thing is how much of this library remains unexplored. Scientists estimate that all the life forms that have ever existed on Earth represent less than a tiny fraction of the genetic possibilities that could theoretically work. It's as if we discovered a few beautiful books in a library so vast that most of its shelves have never been visited. This perspective fills researchers with both humility and wonder. They realize that their work in genetic engineering and synthetic biology represents the first tentative steps by conscious minds into regions of the library that evolution has not yet explored. They are like explorers venturing into uncharted territories of genetic possibility. Some scientists search for genetic sequences that could create new medicines. They look for combinations of letters that might produce proteins capable of fighting diseases in ways that natural proteins never could. Others seek genetic texts that could enable organisms to survive in extreme environments, perhaps even on other planets. The search requires both systematic exploration and intuitive leaps. Computer programs can generate and test millions of genetic sequences, looking for promising combinations. But human creativity and insight often guide the search toward the most interesting regions of this infinite space. As researchers explore deeper into the library of genetic possibilities, they make surprising discoveries. Sometimes sequences that seem completely artificial work better than anything found in nature. Other times, genetic combinations that appear promising in theory fail completely when tested in living cells. These discoveries teach important lessons about the nature of life itself. They reveal that the genetic codes used by existing organisms, while remarkable, are not necessarily the only solutions to life's challenges. There may be entirely different ways to build eyes, hearts, brains, and all the other structures that enable complex life. The exploration also reveals the deep connections between all living things. When scientists create new genetic sequences, they often work best when combined with genetic modules that evolution has already perfected. It's as if the library contains not just individual books, but chapters and paragraphs that can be mixed and matched to create new stories. This modular nature of genetic information suggests that life itself has been collaborating across species and across time to create a vast, interconnected text. Genes that first appeared in ancient bacteria still function in human cells. Genetic circuits that evolved in simple organisms have been elaborated and refined in complex creatures. The more researchers explore the infinite library, the more they appreciate the wisdom embedded in the genetic texts that evolution has already discovered. Even when they can create artificial sequences that work, the natural versions often prove more elegant, more robust, and more beautifully integrated with the complex systems of living cells. This realization fosters a deep respect for the 4-billion-year research project that life has been conducting on Earth. Evolution emerges not as a blind process, but as a patient and creative explorer of genetic possibilities, testing countless combinations and preserving the most successful discoveries. Scientists approach their own exploration of genetic space with this humility. They see themselves not as conquerors of nature, but as junior partners in the ongoing project of discovering what genetic combinations can create beautiful, functional, and beneficial forms of life. The infinite library reminds us that the story of genetics is far from over. We have learned to read some of the books that evolution has discovered. We have begun to edit a few pages and write some new chapters. But the vast majority of genetic possibilities remain unexplored, waiting for curious minds to discover them. Some of these undiscovered genetic texts might hold solutions to humanity's greatest challenges. Others might reveal entirely new ways that matter can organize itself into living systems. Still others might point toward forms of life that we cannot yet even imagine. The journey into this infinite library requires not just technical skill, but wisdom, patience, and respect for the profound mystery of life itself. Each new discovery must be made with careful consideration of its implications for the intricate web of relationships that connect all living things. As you drift toward sleep tonight, consider the vastness of this genetic library that surrounds all of existence. In your dreams, perhaps you will walk through its endless corridors, glimpsing volumes filled with genetic poetry that could create new forms of beauty, new solutions to ancient problems, new chapters in the ever-expanding story of life. The infinite library waits patiently, its books ready to be discovered by minds curious enough to read them and wise enough to use their contents with love.